Recently, Google Research released a multi-agent system called AI Co-Scientist. This system demonstrates how artificial intelligence can serve as a virtual collaborator for scientists, assisting in generating new research hypotheses and accelerating scientific discoveries. This video will provide a detailed analysis of how this system works and its practical application cases. AI Co-Scientist is a multi-agent system built on Google Gemini 2.0 that in today's research field. Researchers face a common challenge: how to maintain a comprehensive grasp of the field amidst rapidly growing scientific literature, while also integrating insights from different disciplines. For example, the CRISPR research by Emmanuelle Charpentier and Jennifer Doudna, who won the 2020 Nobel Prize in Chemistry. Interdisciplinary breakthroughs often require expertise from multiple fields such as microbiology, genetics, and molecular biology. In terms of system architecture, the AI Co-Scientist adopts a method where multiple specialized agents work collaboratively. Each agent undertakes different responsibilities, collectively forming a complete scientific research collaboration process. First, the generation agent will generate a series of potential research hypotheses based on research objectives and existing literature. These hypotheses will then be analyzed in depth by the reflection agent through self-examination and critical thinking. The quality and feasibility of the hypotheses will be improved. This process ensures that the research hypotheses are refined and validated. In the hypothesis optimization phase, the ranking agent evaluates and compares different hypotheses based on multiple dimensions. The evolution agent is responsible for combining high-quality hypothesis features. Through iterative optimization, it generates better research proposals. Meanwhile, the proximity agent continuously monitors and ensures that these hypotheses remain closely aligned with the original research objectives. Finally, the meta-review agent conducts a comprehensive evaluation of the entire process, ensuring that the final research proposal is both innovative and practically feasible. To ensure the quality of the system output, AI Co-Scientist employs an innovative test-time compute expansion technique. The way this technology works is quite interesting. The system allows different agents to play the roles of different scientists and engages in scientific debates through self-play. This is like simulating an academic seminar among real scientists, where each scientist can present their own viewpoints and evaluate and discuss others' opinions. These scientific debates are conducted in the form of a tournament in this process, each research hypothesis is given a score. This scoring system draws on the yellow rating system commonly used in chess, just as chess players receive different ratings based on wins and losses. Research hypotheses also receive corresponding scores based on their performance in the debates. Research data shows an interesting phenomenon. Hypotheses that achieve higher yellow ratings in debates often produce more valuable scientific research outcomes. This indicates that this rating mechanism can effectively identify high-quality research directions. Through this optimization process, similar to evolution, the system can continuously screen and improve research hypotheses, gradually enhancing their quality. Each round of debate helps the system identify better research ideas, just like scientific discovery itself, which is a process of gradual improvement. In practical applications, AI Co-Scientist has already demonstrated its practical value in 3 biomedical fields. 1. Drug repurposing. The system successfully predicted existing drugs that could be used for the treatment of acute myeloid leukemia, AML. Here, drug repurposing refers to discovering new uses for existing drugs. This method is faster and more cost-effective than developing entirely new drugs. Laboratory tests confirmed the system's predictions. These drugs indeed inhibited tumor activity in multiple AML cell lines at clinically relevant concentrations that, in simple terms, they effectively inhibit the growth of leukemia cells, bringing new hope for leukemia patients. 2. Target discovery. In liver fibrosis research, the system has helped scientists make an important discovery that identified some special epigenetic targets, which are regulatory switches that can affect gene expression without altering the DNA sequence. These targets have shown significant antifibrotic activity to validate this discovery, The research team used human-derived liver organoids, like miniature artificial livers cultured in the lab, for experiments, and the results confirmed the system's predictions. This research achievement is about to be officially published by a collaborative team at Stanford University. 3. Antimicrobial resistance. In this study, the system helped unravel a complex puzzle of bacterial evolution that IT successfully explained how bacteria evolved through gene transfer, just like bacteria passing resistance genes to each other, I in particular. It revealed the mechanism of a special structure called capsule-forming phage-inducible chromosomal islands, CFPIC, in different bacterial species. This discovery is particularly interesting because it completely aligns with previously observed but unpublished research results in the lab, further confirming the reliability of the system from another perspective. However, there are still some areas where the AI co-scientist needs improvement. First, the system's literature review capability needs to be strengthened. Currently, the system is not comprehensive enough in processing and integrating a large amount of scientific literature. IT may miss some important research results or key insights. This means that the research hypotheses generated by the system may not fully utilize the existing scientific research accumulation. Second, the factuality checking mechanism needs to be improved. Although the system can generate seemingly reasonable hypotheses, sometimes it may produce speculations that don't fully align with existing scientific knowledge. Establishing a more rigorous fact-checking mechanism can help filter out these potentially problematic hypotheses. Third, the system requires more external tools for cross-validation. Current validation mainly relies on the system's internal evaluation mechanisms. If more professional research tools and databases can be integrated, it will help improve the reliability and practical value of hypotheses. Fourth, there is still room for improvement in the system's auto-evaluation technology. Existing evaluation methods may not fully capture the innovation and practical value of research hypotheses. More refined evaluation criteria and methods need to be developed. Finally, the system also requires larger-scale expert evaluations to verify its effectiveness. Although the initial application results are encouraging, to truly prove the practical value of the system, more long-term usage and feedback from domain experts are needed. These real-world validations are crucial for the further improvement and refinement of the system. In the future, Google plans to open system access to research institutions through the Trusted Tester program. This suggests that AI co-scientists may serve as an auxiliary tool to help researchers accelerate the process of scientific discovery. This project also demonstrates how collaborative, human-centered AI systems can enhance human creativity. It is worth noting that AI co-scientist is positioned as a scientific research assistance tool rather than a replacement. Its goal is to stimulate scientists' creativity by providing new perspectives and hypotheses, rather than replacing human scientists' judgment and intuition. This human-machine collaboration may represent an important direction in the future of scientific research.